I'm sure a lot of you know what OCB is. It's the Ocean Carbon and Biogeochemistry Program. OCB's been around since 2006, and we have a project office based at the Woods Hole Oceanographic Institution that gets funding from NSF Biological and Chemical Oceanography and NASA Ocean Biology and Biogeochemistry. And I came into it in 2007 as a fresh off the boat sediment paleoceanographer. So I'm really excited to be talking about mud today. We have several focus areas. You can see them up in there in the green icons. Pretty broad. Our breadth is really impressive in terms of the scientific scope of the program. It's really just a thriving network of scientists, and we're really happy to have this here. We have an annual solicitation. If any of you are so inspired after this to submit an idea for the community to tackle, we do workshops and we do training activities and smaller scale activities like this, working groups with more of a focused mandate. But it's really a bottom-up program. It's driven by the ideas and enthusiasm of the community, and I'm just here to facilitate. I really work across the agencies and the scientific community to make stuff happen. So that's something I really actually enjoy doing. So if anybody has questions, just come talk to me. I'm happy to chat. I'm also really excited to talk about sediments and biogeochemistry. So you'll see these little signs up posted around. OCB is very, very passionate about being inclusive and people feeling safe at our activities. If you witness anything or you experience anything that makes you uncomfortable, any type of bullying or harassment, please either scan the QR code and you can do an anonymous report of that, or you can come talk to me. This is a really great community, and we haven't had a lot of these reports, but we put this there for your safety and your comfort. So if you witness or see anything, just do that and we will handle it accordingly. So thank you all for being here.
Yeah, so I'm trying to be quick in talking about how we got here. So this started after Wally was a postdoc at JFDL, and we were looking into the model, and that's actually an idea we had after a talk that Sam gave for us there. That it's really become a problem that benthic processes are not represented on system models, or when they are, it tends to be the very simplistic parametrization. And then we started looking into it, and this is a plot that Jessica Liu made. She can't be here today. So just comparing the ESM, so that's one of the newest GFDL Earth system models, bottom oxygen consumption with the newest, the more recent global data set. And you can see that the values are pretty different. The color bars are the same for these two figures here. And we're like, okay, like, so what's going on here? This is clearly not matching. The model's not matching the observations. So if we crank up expert production just as a thought experiment, we still didn't get anywhere near the Jorgensen database, and we ran out of nutrients at the surface. So we're definitely missing some processes here. They need to be included. And then we started a study group at JFDL, just seeing what models are out there, what type of processes they're representing. If we had to dream up a new thing, how would it be? And it was a very fun experiment. And then we're like, okay, let's see what kind of data we would use to try to validate this sort of models and like what's out there. And then things became even more complicated because it was very clear to us that there are several different communities working in the space that are pretty much speaking different languages. So we had a hard time understanding like what the methods are, what the difficulties are, and then sometimes you see, well, this thing is being measured here and it's being measured there, but the methods are very different and you can't compare the data. People's definitions tend to be very different. So a lot of people are like, okay, like that's great that you're interested in sediment dynamics. Are you going to represent this creek that I've been working on for 20 years? And I'm like, well, my model doesn't quite have that sort of resolution. So communication has been very fun because there's a lot of opportunity to learn, and I have definitely learned a lot, but it has also been very challenging. So there's definitely siloed communities working in the space, and we want to try to bridge that. the other thing is that, most of our understanding comes from these places here, right? So this is just looking at some of the global, data sets, more recent data sets. And I just did a comparison of how much, of it comes from the South Atlantic, so down to Antarctica. So like all of this region here, I mean, the point in, you don't even have to, didn't even have to do that calculation, because there's just like a few points here and there. While you have a lot of it in Europe and in the US. But looking at the Jorgensen for total oxygen uptake, 6.8% from the southern, South Atlantic. And the Ben Bioden, so that's just like, benthic fauna, 2.3%, although it looks like more points. Like 70%, 80% of all the values from the South Atlantic come from this one cruise here. So it's not a lot of data. So when we talk about global data sets, is it really global? Like, are there any processes that we're missing by not representing, not having more spatial coverage? And if we want to increase, spatial temporal coverage, what variables do we want? So a lot of times we get asked, like, what do you want from observationalists? And I'm like, I don't know. My whole career has just been getting whatever I can get my hands on, so I'm not really used to that kind of request. So what we're trying to get here is like facilitate communication and data sharing between coastal and global biogeochemical modelers and observationalists, establish some shared data protocols, including some common definitions and names, units. So when we say coastal, what exactly are we talking about here? Develop data products for model development. So can we agree on some things that go together and how they go together, so that we could put them, in a uniform spatial temporal distribution that people can actually use for model purposes, and develop recommendations and future directions in observations and models. We're aware that we have to start, whatever it is that we come up, it's not going to represent everything we want to, but these things are not represented at all sometimes, and it's better to have something. So some of the questions for this workshop specifically, these are more the motivations to have BECS. And some of the questions that we want to answer on this workshop is just what are some terms and definitions that we need to agree on? What processes do we need to understand better, and what data do we need for that? And how do we bridge across spatial and temporal scales? So if you're doing a 1D model to study some coastal environment, that's definitely going to be a very different model than what an Earth system model is going to need. And what are some opportunities to increase data coverage, and how can we create strategies that are actually accessible for folks outside of the US and Europe, and opportunities for collaboration. So what we want to get to here, and I'm just putting this here so everyone can start thinking about it, is one of the deliverables we want is a perspective paper. So the way we thought about it so far is just an introduction, review of processes, some questions of scales and differences in definitions. And what we want to is identify 5 major challenges and opportunities. So we know there's a lot that needs to be done, but instead of just thinking of like what the problems are, like how can we move forward also? Be a little more positive. So just, we should think about what would be a target. I did some digging on like what are some of the some journals that we can start thinking about. So yeah, just so people can think about it. I said this yesterday, but just to reiterate, so we're gonna have a few talks, and then we're gonna have breakout groups. So there's a paper on the board in the back here. The breakout groups have been randomly generated. So we're gonna have 2 here, and the other groups in the other rooms here. And the idea is to have a 1-hour discussion. And instead of a report back, everybody comes here and tries to summarize an hour of discussion in 5 minutes. We have these boards here, so feel free to use it. And the idea is that we're going to have a mini poster session after the discussion, so we can go people from the different groups can go to the other groups and actually have more of a discussion. I'll try to be as organized as I can in keeping track of everything. So on Thursday, we can actually go through a document as a group and think about what do we do as a community moving forward. So,
well, thanks everyone for being here. I have this distinguished job of being moderator, which is really just gonna stand here and say our next speaker is. And then I'll be taking notes and stuff too. But I do just want to say how excited I am to be here today, and thank you, Cristina and Heather and everyone from OCB for all their efforts, because it's not easy. I just love sediment so much that this is just like really the best setup for the next few days. So yeah, our next speaker doesn't need really any introduction. So David Burdige, please come down and talk to us.
Thanks.
Yeah.
So when they asked me to give this talk, I thought, I teach a whole graduate course in this. So I gotta cram it into 20 minutes. And also I didn't want to step on the toes of the other speakers. So I put something together. It's going to be a lot about oxygen, although I didn't really anticipate it. That would be the case when I started to work on it. So I just threw this up. This is a review chapter. I think it's been published. I'm not sure. You never know with Elsevier. But when it does get published and they do send me a PDF, if people want copies, just let me know. Because I think it's fine. I think it's pretty good. So, and it's got a lot of the stuff that should be relevant and interesting. So this is a picture that came from that book, and it's I'm going to use this as a framework for a lot of the talk with regard to the processes that we're interested in. And I'm not going to focus on all of some of the details here, because there are other talks that are going to present that stuff. But what the point I wanted to make with this was that when you have suboxic or anoxic remineralization, you end up producing reduced end products like methane or sulfide or iron or manganese. And these can then get used by chemolithotrophic reactions, because the way I like to think of it is that some of the energy that was stored in the organic matter wasn't utilized by the anaerobes, and so it ends up in these reduced end products. And so that's what. Anyway, that's what some of the horizontal red arrows are meant to indicate in this figure. And the reason that oxygen becomes important in this regard is that in many systems, there's a really tight coupling between the production of these reduced end products and their oxidation. And so the net result is which one of these is the arrow?
the forward or?
No, the pointer.
Oh, yeah.
Oh, there we go. Okay. So the point is that if you're sort of staring at the sediment from the top down, what you could argue you're seeing is something that looks like this in terms of oxygen consumption, even though much of the carbon may be degraded by bacterial sulfate reduction. And so this is a very simple system. And we now know that oxygen is not the only oxidant, that nitrate can be important in a lot of systems and even metals like manganese and iron, but this is a good example to use. And so I just threw this in because I'm sure someone's going to talk at some point about cable bacteria. There obviously this process is, so yeah, this process is mediated by cable bacteria. And I'm sure someone will tell you more about that later on. But these kind of redox couplings can be even more complicated. This is a picture that I originally stole from Bob Aller from a paper of his, about how you can couple sulfate and manganese and oxygen together. And again, the important point is that if you're staring at this system from the top, you could measure oxygen uptake, and it would be a reasonably good proxy for total benthic respiration, even though virtually, well, not virtually, but even though a small fraction of the respiration in the sediments was not driven by aerobic respiration. So this is kind of illustrates partially that point, that if you look at respiration in sediments and you go from the coastal region out to the deep sea, out in the deep sea, most of the respiration is aerobic. But when you get into the coastal regime, most of the respiration in the sediments is not being directly mediated by aerobes. This is a figure that came out of, so we saw some of the data from that Jorgensen paper on a 3D contour. But what I did is I took, this is their compilation of all the oxygen uptake data that they had put together, and I just made the point here that in the deep sea, where the rates of oxygen uptake are low, it's all aerobic respiration. But as you move into the coastal regime, the oxygen uptake increases by 3 orders of magnitude, 2 or 3 orders of magnitude. But the oxygen uptake is largely driven by oxidation of reduced end products like sulfide. And I think in that same Jorgensen paper, or maybe another Jorgensen paper, he estimates, or they estimate, that 50% of the total oxygen uptake in coastal sediments is due to sulfide oxidation, and even when you get out onto the continental margin, it's 25%. So that's perhaps not surprising, because if you look at the relative importance of aerobic respiration to carbon oxidation, and then you look at it for sulfate reduction, they're sort of pseudo, I don't want to call them mirror images, but they're sort of mirror images in a sense. There's essentially very little sulfate reduction out here in the deep sea, and it's all sulfate reduction inshore, and the reverse for aerobic respiration. Okay, so this is a figure that I put together. So all the little red dots are estimates of carbon oxidation rates. Now, some of them may actually have been derived from data points that are shown here, but a lot of them are independent estimates based on diagenetic models, rate incubation studies, benthic lander determinations, and whatnot. And the red line here is just my attempt to sort of brute force a couple of straight lines through the data. I don't remember why I did that, but I did that for some reason. And then the blue line is essentially the Jorgensen et al. Fit to the total oxygen uptake data from here. So I basically took this line and put it here, okay? And the point of this is to sort of make the observation that at a kind of a first order level, this very, very oversimplified view of carbon respiration in sediments is not a bad way to estimate at least integrated rates of processes in particularly sediment environments. And the idea is that the sense is that the blue line, granted it's a log-log plot, so as you know, log-log plots hide a lot of sins. It's a pretty good agreement, okay? But that's it. Life is never that easy, and particularly when you get into the coastal realm, there are a lot of things that we do need to worry about that probably are important. And so one of the ones that has interested me off and on over the years is pyrite burial, because if you bury pyrite, then you decouple this the oxidation and reduction here. And this can be important in coastal systems because of sometimes the pyrite burial can be seasonal. So you can bury pyrite in the winter when rates are slow, and then when things speed up, you'll oxidize it. Okay. Or did I get that backwards? I think I got it backwards, yeah. But anyway, there's seasonal storage. And so your oxygen, for example, oxygen uptake measurements are not going to work to estimate carbon respiration in the sediments unless you integrate over the appropriate seasonal time scale or something. And then the other one that's probably important to a lot of people here is denitrification, because when you make N2 gas, it goes away and it also acts to decouple. And so depending on where you're working and what you're doing, these could be important issues that important aspects of the sediment biogeochemistry that you need to worry about. Okay, and this is just a table. This is a table that I put together to illustrate this point on a global perspective, that here is an estimate of global sulfate reduction rates. You know, inject sulfur 35 into a core and measure sulfate reduction. And if you compare that number to a couple of different estimates of pyrite burial, and then you just take the ratio of the two, you see that the sulfur burial efficiency is, you know, anywhere from a couple of percent to 10% or so on a global basis. Again, this is sort of a broad scale view at 20,000 feet, because we know that there are lots of places, particularly in the coastal regime, where you get very, very high sulfur burial efficiency. The one that I know and love the best is Cape Lookout Bight, but certainly some of the original sites that the folks at Yale were studying in Long Island Sound have these very high rates of burial efficiency, and it's a combination of high sedimentation rate, high sulfate reduction rates, but to a large extent, it's oftentimes just a lack of bioturbation and bioirrigation. So if you don't get the oxygen in, you can't oxidize the pyrite and it gets buried. So, okay, going to change a little bit and talk about organic matter. This is again a highly simplified view of how we like to think about carbon degradation in sediments, that you start with complex organic matter, and by processes that you might think of as either hydrolysis or oxidative cleavage, you start to break it down. Ultimately, you end up with a limited number of small fermentation end products, which then ultimately get fed in to these anaerobic processes. We know this quite well for sulfate reduction, methanogenesis. We don't know this nearly as well for the metal oxide reduction reactions or denitrification, but I think there is enough anecdotal evidence to suggest that this is probably a reasonable model. The important point is, again, if you're looking at this from a great distance, there's a very tight coupling along the way here. And that's why in a lot of models, you can write equations like this that seem to imply that a sulfate reducer chews on some mythical CH2O and makes bicarbonate and sulfide. They don't. They use acetate, they use hydrogen. But because these processes are so coupled, you can write it that way. If you want to be more a little bit go into a little bit more detail, this is sort of a conceptual model that we've worked with over the years for the processes that may be impacting carbon cycling in particularly anoxic sediments. Most of the carbon flow that I just talked about a minute ago sort of goes in this direction from POM, high molecular weight, labile stuff, into what we refer to as monomeric low molecular weight DOM, so it's acetate. I mean, I like to tell my students all roads lead to acetate in anoxic sediments. It's really, they lead to acetate and hydrogen, but all roads lead to acetate. But there are other things that go on that along the way you produce dissolved organic matter that is refractory, and again, refractory is always in the eye of the beholder, because what's refractory to you may be very labile to someone else if they've got a million years to chomp on it. But in some systems, this is refractory. Studies, incubation studies that various people have done have shown that you can produce low molecular weight refractory DOM in ways that are consistent with this model. The other important point is that much of this low molecular weight refractory DOM is largely uncharacterized, certainly at the molecular level. Now, with new techniques like Fourier transform, let's see if I get it right, FTICR-MS, the alphabet soup toy of organic geochemists. You can get a lot more information about what these compounds look like. So it is, we are beginning to have a better sense of what this material is, what it looks like. The thing that may be important to people here is that a lot of this material may contribute to the benthic flux of dissolved organic carbon. And if you start to think about export or outwelling of DOC from coastal settings, what some people refer to as blue carbon export, if you're making this kind of refractory material in sediments and exporting it, it may represent a way of sequestering carbon in one way, shape, or form. Okay. So just one last point. When we start to think about what may be the rate limiting step in this decomposition, we oftentimes think it's this initial, what I sometimes call depolymerization. And this is in a sense where you could argue that oxygen might be important because there are, in my sort of simple view of the world, there are two ways you can sort of break down large polymers. One is by hydrolysis, like proteins, peptides, and carbohydrates. And that can be done both in the presence and absence of oxygen. And then the other way is something that I like to think of as oxidative cleavage, which is something that is more, which requires in one way, shape, or form oxygen, whether directly or indirectly by producing free radicals that help to break down things like, lignin molecules or complex aromatics or whatnot. So this is where oxygen becomes very important. Benthic macrofauna are really important here, as many of you know. They lead to what I like to think of as redox oscillations, which means that their activity periodically pumps oxygen into the sediments one way or another. The same thing happens in vegetated sediments. Salt marsh plants, seagrasses also pump a fraction of their oxygen below ground and can stimulate this oxygen input. The oxygen effects are also a little indirect, because when you start to pump oxygen into sediments, you stimulate metal redox cycling, iron and manganese, and these oxides are good at breaking down refractory compounds. And something that I think is potentially really important is this redox cycling will produce something, can produce something that's known as Fenton, the Fenton reagent, which is basically iron 2 and peroxide. And that is really good at breaking down labile and refractory organic matter. And it's a very potent oxidant. And then I put this in here because iron is really, the more I think about it, the more I used to say that manganese was my favorite element, but iron's maybe taken over because iron, a lot of people, and there's this classic paper by Lalonde et al. On the role that iron plays in promoting organic matter preservation. I would argue that iron and manganese are also important in promoting remineralization. And I think it's not that they're wrong and we're right or whatever. I think it's a time scale and a system consideration that in some cases there are situations where iron is indeed going to promote preservation, but it's equally important in other systems for degradation. Whether this is one of the big 5 problems, I mean, I would argue it might be, but we'll have to all be the judge of that. So, that's it. I put this in just because this is one of my favorite pictures. It's really you can actually learn a lot about sediment biogeochemistry if you stare at this picture and ask questions like, what are these little bubbles here? Why is this brown and why is this black? So, anyway.
Great. We're
I'm
gonna
done.
hold questions just so
Yeah.
that we are on time and
Sure.
have lots of discussion afterwards. So next up. Thank you for that. That's a great way to kick it off. And probably all of us want your PDF. So next up is Talia Evans from UC Santa Barbara, who's going to talk to us about sending their redox ladder. Great title.
Wasn't mine, actually.
Yeah.
I'm just signing this. Anyways, everybody, thank you for having me. I'm really flattered to be able to get a presentation. I figured most people kind of know this already and also got a great intro to it. So I'm gonna go kind of quickly through it and then go out on a limb on some stuff. So it doesn't make sense. It runs like 16 to 18 minutes, so feel free to interrupt me with a question. There, okay, it does work. So first part is just hitting the redox ladder, of course. Then we're gonna take a little digression into like thermodynamics, kinetics, and microbes and the role in that. Move into thinking about redox systems being as like stable and unstable states, and scale that up into thinking about like capacity and resilience for redox. So that's sort of where we're going, and that's the sort of the limb I'm gonna go out on here. So first, redox ladder stuff. We just saw this reaction in a different way. We have organic matter and oxygen, and it's being used to break down organic matter to create CO2 and other nutrients released, right? So the question is, why is oxygen what's being used here? And that can be, sorry, am I in the way? Okay. So what you can do is oxygen allows you to sort of pull that apart into half reactions. And you can see that there's these 2 combinations of organic matter breaking down, and oxygen being used as a terminal electron acceptor and taking those electrons. And that opens up the ability for other ones to take electrons too. And each of these have their own electrochemical potential, right? You put those together, you get a reaction. That's going to the delta G of that reaction. So pulling this out into Gibbs space, the Gibbs free energy of the reaction is some combination of some standard state plus the amount of that reactant, right? So oxygen is put energetically, which is the first part, the delta G standard state, and it's also abundant. It's the second part. Right? So that's how that sort of works together. So this figure here is we're going to have the sort of profile going down, which you've seen a little bit in the last talk. So all the processes are going to be named. Right? And the point here is that there are now, if you run out of oxygen, you go into like nitrate reduction. These are canonically dictated by the Gibbs free energy of reaction. So a lower energy is more spontaneous, you get more energy out of it in theory. So first is oxygen being the most favorable, and then nitrate you see is a little bit more positive. So what happens next? And then you drop the whole ladder down, you've got manganese, iron, sulfate, methanogenesis, etc. As the numbers start to go up, right? That's sort of the foundation for why we have this redox ladder in theory. Okay. And so here's a better picture. We also have the product. So on your website, we have the reactants, so what the electron acceptors are. And on the right side, we have, well, in most places, what the products of the reactions are. So we're making manganese, iron 2, sulfide, methane, nitrite is an intermediate there. It's also making nitrogen gas and making water, which is not shown for the top one. So the main points I want to drive home is like, when you like textbook wise, like redox ladder is that you have these delta G's, which fixes this order as you go down, right? That's what controls that. You switch metabolisms when one is no longer favorable, right? When it's so low abundance, that's the abundance. The next one has a lower delta G, and therefore it's used. So that's what the switching point that is. If you look in the textbook or thermodynamics, what would it say, right? And then we got a great talk about how the overlap of some reduced products with some oxidized reactants can cause chemoautotrophy. Let's start with the trophy before. Same story. Great. Thank you so much. And I have my example here is nitrate and iron 2. Cool. Any questions? Good. Cool. Awesome. Fun stuff. So I want to really hit home that microbes drive most of these reactions. Right? They are catalyzing most of them. That there are some that abiotically can also occur, right? But in many cases, particularly if you're in a water column, microbes are very important here. And I want to make the bold statements that the energy that a microbe gets out of this reaction is linked to, but not completely described by the Gibbs free energy of a reaction at its given temperature and pressure. Right. So here's sort of one of your classic Gibbs free energy diagrams. We have the energy on the y-axis, right? In this case, we have the reactants and it going to a lower energy state, which is the products. That's energy of the reaction is the delta there. And there are two hills, a blue and a red, right? That's sort of the activation energy barrier. They get from the reactants to the products. And the role of the catalyst or the microbe in this case is it makes it easier to get over, reaction happens faster, right? But if you're a microbe, you don't just get the energy out in that reaction, right? Because you have to still make up the hill. So there is some energy loss due to overcoming the activation energy barrier. There's also energy loss to the fact that the enzyme isn't perfectly efficient, right? As well as actually to build the enzyme and the transporters, etc. So there's sort of this things about like yields and efficiencies. That means that delta G is not always the like controlling factor, but there's these things that like pull out of that energy. Where I'm going with this is thinking less about this as like a single reaction, but more as this ability to supply power, right? Because microbes like exist over time, and the sort of turnover rates, right? The ability to fuel that microbe. And if you happen to have a reaction that's very slow in nature, say we have like ammonia oxidation, right? If you just have ammonia and oxygen, but it's completely sterile, the activation barrier abiotically is too high. But with the presence of some catalysts, this reaction can occur. That's the sort of case I'm showing here, where when you add a little bit of catalyst, right, the reaction rate of that sort of substrate system slowly increases until you hit the point of mass transportation. So we're moving from a sort of slow reaction to a one where you just hit that substrate, you hit it and it immediately turns over. So like oxygen respiration, things are really rapid consumption of like materials. You can switch from the sort of like kinetic control where it's limited by the ability to catalyze it to there is so much enzyme, so much bugs there that they just immediately take it out. And that's sort of a mass transfer control. That's what limitations. So that is an interesting thing you can see if you just look at various data as a thermodynamic favorable reactants. Like this is a reactant that could be on a receptor, aren't being used, and they persist in more reducing conditions. Whereas things that are oxidized products can also, reduced products can also persist in more oxidizing conditions. So where am I going with this? As a tale of two terminal electron centers, right? Once again, we have this redox ladder here, and I'm highlighting iodate reduction as this couple and iron reduction as this couple. Okay, you'll note that iodate is right below oxygen, right? So once oxygen is used, thermodynamically, iodate should be the one that kicks in. And after a while, once you deplete your nitrogen cycle and some of your other things I'm not going to talk about, you switch to iron, which people here know more, right? So background, that's to talk about iodate a little bit, is that iodate reduction produces iodide. It's quite stable in the water column. And rule of thumb is about 450 nanomolar iodide, so that's sort of your inventory space, right? If you have completely oxidized, it's 450. If it's completely reduced, it's 450 iodide, right? Iron reduction produces iron 2. That's remember, it's not as stable. But since iron is mostly in the sediments, there's not as good of an inventory in the water, right? It's basically just how close you are to terrestrial system. So in the summer of 2021, we went to the Oregon Shelf during summer hypoxia. And you can see here's a profile of oxygen in the system. You see at the surface, there's saturated oxygen, it's about 350 micromole per kilogram. And near the sediments in gray, sediment interface, about 200 meters, it is hypoxic. It's about 50 micromole per kilogram. Right, so we have high oxygen. Going lower, consumption of oxygen and reducing the sediments. This question to the audience. What do you think the iron 2 and iodide profiles? Those are both the products of those two reactions, right? What do you think the iron 2 profile look like? You have a reducing sediments, a hypoxic water column movements. Yeah, so it'll be higher at the
High
bottom.
at the bottom and it goes to zero very quickly.
Yes, cool. What about the iodide? The same as the iron because it's also a redox compound and it's thermically more favorable than iron.
Increase in the decrease
So it's higher in surface, decreases at,
as the iron increases.
okay.
Like me.
So any other guesses?
I would say maybe lower on the sulfate, increase in the sulfate, and then decrease.
You want to see?
Okay.
So the iron 2, everybody was got right? Sediments provide iron 2 into the water column, and then it's oxidized, right? The iodide profile, however, is quite different. There is surface enrichment of iodide due to photochemistry, which is, I didn't introduce, but that sort of thing happens. But below that, you'll see that there's only about 10 to 70 nanomolar iodide, which is much less than the 450 nanomolar total inventory of iodates, right? There is an increase near the benthic system, right? So what you'd expect there is you're thinking about stuff breaking down, right? Purely from a thermodynamic lens, you'd expect there to be a complete reduction of iodates. It's more energetically favorable to produce iodates than iron. And if it's purely sediments are reducing because they're reducing iron. Possibly they should be reducing all of their iodate before their iron, right? But they're not. So you only see a small fraction of enrichment of iodide, the product of iodate reduction, in the water column. And I could even argue that there is always a little bit of iodide in organic matter, right? There's always a little bit of iron in organic matter. Possibly the enrichment we see near the continental margin is just due to diagenesis and breakdown of iodide in organic matter. Could not even be iodate reduction itself, not that how it may not active. Right? So the big point here is that just because it's thermically favorable to do, doesn't mean it's going to happen. Right? So now thinking starting to go out on that limb. Okay, so reduxes with states. And I'm going to present some data from Sebastian Van. Yeah, what's that?
In your previous profile, what did your dissolved manganese?
Great question. I don't have the data on me.
Is it iodate reduction also coupled with manganese?
Yes. It is.
Okay.
What does this data look like? There was that much manganese actually. We didn't get that much dissolved manganese compared to this. Yeah. I think there, yeah. I'd also argue that this temperature iodate reduction is much faster than the manganese particle catalyst stuff that happens on it. At least at seawater features. But it's a good point, and that's actually something I'm trying to talk a little bit more. Because we don't have very good data for that in like seawater. It's much more terrestrial. So switching to thinking about redox as states. So we have these nearby salt marshes, right? Seen in bottom figure, right? Pretty close together, right? And they have very different chemistry. So Some of them are predominantly iron reducing, some of them are sulfate reducing. It's sort of cool. You can see that in some of them we have minimal iron accumulation and significant sulfate depletion and sulfate accumulation. In others, there's some iron accumulation and not much sulfate depletion, not much sulfite until you get to the bottom little bit, right? And the study, as people here probably are very aware of, is that the bioturbation is correlated with iron reducing conditions, and that the sulfide production probably poisons and bioturbators. Where I'm going with this, though, is the question is, what causes things to switch between these systems? Can you make one of these salt ponds, salt marshes, into from iron reducing to sulfate reducing, and vice versa, right? How much of perturbation do you need to do that? And that's sort of to think about a different Van de Velde paper, where these are, once again with this energy diagram, right, where low is stable, as these are different stable states that you can move between. And if you add more organic matter, you drive your reaction further. You could get into like sulfate reduction states. Also, if you add a bunch of sulfide, you could poison these at bioturbate, and then, right? Or on the other hand, you add more iron, you pull the sulfide out of the system, you now have more iron to reduce, or you just oxygenate it, right? Could also work. Possible things that can switch between these things that are like states now. Now, all the way up there is climate change causes these sort of redox perturbations. This is a accumulation of data from the Oregon continental shelf again. From 2005, there is oxygen on the x-axis and depth on the y-axis. As it's proteoxic at the surface and goes down, there's a little inset map there showing where the data points are from. And the big point of this figure is that in 2006, we saw the immersion of anoxia in the California current system off the Oregon continental shelf, right? Had a very big impact on the fisheries there. As the water column near the bottom, we had some sulfidic bottom water, I believe. So if we think about this one, see in sort of states where we have this oxide state, we're moving away from that biogeochemical baseline. Can we think about this from like a redox perspective, or like a resilience perspective? We have a pretty good understanding of how redox perturbations and systems respond in sort of carbon system space, because we've done a lot of work on the ocean carbon system, right? So if you change the pH, we have alkalinity, we have buffering capacity. Understand how a pH change will respond in the system, right? How much pH, how much acid added, change the pH, etc. Right? One thing this paper by Burdige and Wally talks about is, can we develop a similar perspective for redox? Right? So if you have a freshwater versus a saltwater system, which is the comparison, there are different inventories of electron acceptors, right? And there's sort of buffering capacity set by these compounds. And while, for example, for the freshwater has more nitrates and it's more iron, so it's harder to get the sulfate reducing in the freshwater. It's much easier to methanogenesis because there's so much sulfate in the ocean, right? And can we conceptualize this way? I don't know if we can. There are some different downsides because the redox here is facilitated by the microbes, as hit home earlier, and that's often to do these sort of metabolic changes between terminal receptors, requires metabolic shifts too, in many cases, or new enzymes. And so not sure it'll work, but could be a very interesting direction to go in. And the reason why I'm bringing this up is because you could try to ask questions about how long these ecosystems can persist under deoxygenation before the biogeochemical baselines change, right? So have like, those crabs, you need to have oxygen, etc. And if it goes anoxic, those can die. And then, but upon, that gets into this sort of resistance and resilience framework that I'm not very much of an ecologist person, but we sort of see this sort of system property, which we could say is terminal temperature, etc. And after some disturbance in the red line, it switches into a new stable state, or it can recover, right? There's some sort of lag time and some sort of recovery time from this perturbation. So then as this paper posits is thinking about that, for example, being soils when it rains or when it floods, the effusion of oxygen is limited into the soil there, and you switch from apparently safe respiration to, in this case, they're fermenting, they're making anagenesis, right? And then there's some lag time there, and also some recovery to capacity oxic state. So the first question is, how long can this ecosystem persist with deoxygenation with this redox perturbation? The second is, how quickly can the oxic conditions reassert themselves? Get back to that biogeochemical baseline of oxic, which is then different than the ecosystem recovering, right? Because you need to have an oxic ecosystem, but you can have many oxic ecosystems. Not necessarily the, full crab fish one. Which brings me to conclusion slides, which is most of that. Ramps is not like the complete dictator of active metabolites. There are other things that are important, right? And there's energy costs, and thinking that's more like an engineering framework, there is like losses due to transport and activation barriers. I also really want to hit home on the microbes, really important. If you will, a lot of this redox happens because of the microbes, and they speed up abiotic processes significantly. And that's the sort of limb I went on stability and resilience is sort of something that people are thinking about just based on some of the papers I put up, but it's still in development and an interesting thing to think about with regards to climate change. Okay. So for big acknowledgements for the people who are putting me to come here, pay for the flight, et cetera, see what funded the iodine work and et cetera, and all the stuff that I sort of just showed as speakers that I didn't collect at all.
Okay, so next up we have Sairah Malkin, who's online joining us.
Hi, everyone. Can you hear me?
Yep, loud and clear.
Oh, wonderful.
I'll have your slides up in just a sec.
Great. Well, thank you very much. Thank you to the organizers. Thank you for everyone for being here. I really appreciate the opportunity to participate in this. And I deeply regret that I can't be there in person. So I hope that will not be the case in the future. Thank you very much, May, for running the slides here so that I can look at my notes so that this lecture, I'm aiming for 20 minutes, but it certainly is condensed from, about 90-minute lectures. Plus, I wanted to give everyone in the room something interesting to think about too, if you're already very familiar with all this information. So I'm Sairah Malkin. I'm at the University of Maryland Center for Environmental Sciences at the Horn Point Lab. And what I'm showing here is, you don't need the details here, but it's just some unpublished data from our lab showing biogeochemical profiles from the Chesapeake Bay down core alongside a barcoded, so a 16S gene record of the microbes that are following down through those depths. My point here will be the high level of structure that's available in such data sets may not be yet at a level where we can that we can fully utilize in our biogeochemistry models. But there is a lot of information that's available there that we hope to be able to tease apart. So a side note is that in 2021, the bacterial and archaeal phyla underwent major name changes. I have trouble remembering these. I'm largely using the preceding names, the historical names, which is mostly what's in the literature now. So why might we care about microbes in the seed floor? As we've heard, they're the main catalysts that increase the rates of transformations by orders of magnitude. Microorganisms, to a large extent, therefore, regulate the rates at which elements are cycled in the seafloor, and so they can influence the forms that accumulate in the environment. Microbial communities are extremely complex, and so it might seem like any reaction is possible. But in fact, microorganisms themselves have evolved very specific metabolic pathways, many of them largely conserved, which allows us to use their gene records to inform what processes are actually occurring. So in that way, microbes also may direct the pathways of elemental transformations in the environment. So next slide. So this figure is widely used. You've probably seen it a million times. It's just to show that the deep subsurface is the major area where you find the largest number of bacteria and archaea. Admittedly, that is because those pools of carbon turn over very slowly, but it is a major area for them. So let's just go to the next slide. So geochemical cycles follow the basic laws of thermodynamics, as we have just heard. They catalyze reactions that are on net thermodynamically favorable. They proceed toward the state of maximal entropy and the most stable mineral forms. Microorganisms themselves, though, possess an enormous diversity of catalytic capability, and our understanding and knowledge of that is continuously expanding. One of the ways we can make sense of that, to organize it, is with this standardized nomenclature. These are the 3 basic needs of microbes. They need an energy source, they need an electron donor, and they need a carbon source. Very often, those electron donors and carbon sources are one and the same, but they don't necessarily need to be. And our naming conventions of those dictate where those things are coming from. So heterotrophy is based on the oxidation of organic matter, and yeah, and we've talked about chemoautotrophy here too. Okay, next slide. So what shapes the distribution of microbes and their genetic repertoire through sediments? I'm going to give you 3 slides on that question. This paper here by Petro et al. From 2017 was a meta-analysis of 16S gene sequence archives. And what you're looking at there on the y-axis is the depth interval in meters in the sediment, so not of the water column. And at the time, there were 9 studies where there was enough data available for them to perform this meta-analysis. So what we're looking at are hundreds to thousands of meters deep in the seafloor. And what it shows, okay, so my organization for the rest of the talk after these next 3 slides is that we'll walk down the redox ladder, and I'll talk about the different biogeochemical processes, but from a microbial centric point of view. But what I want to point out is that the microbial communities strongly reflect also the carbon concentration reactivity available at each of those depths through the sediment column. So the first 2 panels, A and B, are showing different metrics of biodiversity, and they're strongly decreasing with depth. And on the panel C, what we're seeing is that the cell numbers themselves are also strongly decreasing. And this is largely reflective of the amount of carbon and its reactivity that's available for the microbes to persist on. The electron acceptors also strongly influence the microbial communities and their genetic abilities at those depths. And those 2 things strongly interact. So if you have more reactive carbon available in the oxic zone versus the if you have a very high sedimentation rate and you have highly reactive organic carbon, but in anoxic depths, that will also affect the types of communities that are there, and those can have nonlinear feedbacks. I mentioned also hydrogen concentration. David Burdige, you talked a bit about acetate. Those are highly connected. I mentioned hydrogen concentrations as important, especially at those sulfate reducing and methanogenic depths. And I'll mention that again on my last slide. There's a myriad of other factors that influence the microbial community structure, including pH, temperature, salinity, pressure at high pressures, and sulfide concentrations. Okay, next slide. So I gave a talk last year about microbes and sediments to a group of phytoplankton biologists, and I realized at the end that they were all thinking of the sediment as a paleo record of microbes. So I included that here. So are microbes basically indicators of the time at which they were deposited on the sea floor, or are they in fact living and potentially evolving at the depths that they are found? The answer to that is largely the microbial communities largely reflect the sediment conditions at which they're found. So at the surface, in that bioturbated zone, there may be evidence of a lot of the microbes that have come from the water column. And these include the bacteria that are attached, associated with sinking particles. They tend to be among the Chloroflexii and Planktomycetes that are really well adapted to attaching to particles, as well as cyanobacteria that are responding to the light in the surface waters. And those will persist through the surface sediments and may even be preferentially preserved. However, at deeper depths, the microbes that are there, this is a very nice paper from 2018 by Tortilla et al. And what it's showing is that the extracellular DNA, the microbial community composition as identified by the extracellular DNA is very similar to the intracellular DNA. meaning that the DNA pool is turning over even at depths. So that's to answer that for the most part, unless you're talking about spores, the microbial community at depth is reflective of those conditions. Okay, next slide. What I haven't told you yet is the actual generation time. So with that bar on figure at the very top, my point was that they were turning over very slowly. I just haven't included the figure here. But if we think of E. coli, say, growing in a lab, they might be growing and turning over at rates of minutes, hours. That's not the case in sediments. And at the surface sediments, we're looking at pools that are turning over weeks, months, years. And as we move down through the sediment column with ever decreasing energetic yields for their metabolism, we're looking at turnover times of the biomass in the order of 10 years. So 10 years or more. So what we're looking at then are at, time scales of, say, 500 years of deposition, we're looking at the number of generation times being on the order of, say, 200 or so generations. So one question, I did pose this right before, are microbes evolving to the depths that they're at? Well, over the course of, say, the Holocene, there's just not enough time. There hasn't been enough genetic generation time for new mutations to arise and spread through population. So largely, the microbes that are there tend to be filtered from the microbes that were present, though potentially at very low abundance, at the bottom, say, of the bioturbation zone. So there is very strong filtering selection, but not a lot of further evolution. Of course, there is a very nice Bill Orsi paper that did grow up some microbes that were more like on the order of millions of years and found that there was genetic drift. There was differences in those microbes from their modern counterparts found at the surface. But in general, what we see at depth tends to be reflective of those that can survive at that depth that had already been a part of the microbial community at the surface. This paper that I'm showing here shows some of that meta-analysis, some of the major groups that tend to persist through depth. You can see the major changes that are occurring through the most common 20 taxonomic groups that they found. Okay, next slide. Okay, so this slide here, 12 minutes here. So it's most certainly true that bringing microbes into culture in the lab and examining what they're capable of doing strongly augments our understanding of what can possibly be occurring in the environment and in our ability to interpret the genetic record that is in the environment. But is there value also in the information at our current ability to interpret it in the microbial records that we find, typically of DNA, which is what most of the repositories would have. Again, this is just some 16S data from my lab. This is from a collaboration with Caroline Slump that is currently unpublished. These are 5 stations from the coast of California that have different bottom water oxygen and nitrate concentrations. And you don't need to know what's in here at all, but the important point is that it's strongly structured. So if we think that there is a strong reasons for that structure, then there's a potential that it can help us in our understanding and our inference of biogeochemical processes. What I also want to convey is, although microbes are the catalysts for a lot of the important reactions, in many ways we can think of the community composition, their genetic repertoire, as the response to ultimate forcing factors. However, a high proportion of the lineages that we do see at depth are lacking cultivated representation. So this is the microbial dark matter. It is very hard to interpret the information. So the information that we have is steadily improving, but it still remains highly incomplete. Okay, next slide. Oh, next slide. Yeah. This is just to say that now what I'll do is I'll go down the redox ladder and I'll show some of those major pathways. And at some of the steps, I'll talk about the reoxidation. Okay, we can go to the next. Okay, so heterotrophic aerobic respiration is the most oxygen is the most energetically available electron acceptor. Microbes that use oxygen tend to have the ability to grow very quickly. They tend to be motile. You can actually see that under, say, a microscope slide. If you had a slide that was anaerobic at one end, and if you watch the oxygen front, you would see a bunch of motile bacteria there. They tend to be able to use a diversity of organic carbon substrates or reduced mineral compounds. And many of them are able to catalyze the full mineralization of complex organic matter all the way to CO2 with extracellular hydrolysis in there. They tend to require the specialized ability to cope with oxidative stress. So they tend to have genes associated with that. So these dualities, high energy yield, along with the stressful environments, tends to make oxygen a fairly strong barrier, where oxygen and nitrate using microbes tend to not be able to cross over into anaerobic environments and vice versa. The final enzyme in aerobic respiration tends to be highly conserved. The major groups are these heme, copper, oxygen reductases. There are different varieties that have evolved along different lineages. So they can be used for phylogenetic analysis. And additionally, there's a complementary one, cytochrome quinol oxidases, but they tend to also be used for multiple functions, including oxidative stress. There are additionally, in this key reference here, they found an additional 720 genes that were involved in aerobic respiration. The aerobic respiration genes then tend to be not helpful for estimating rates of aerobic respiration, but they can be good at identifying aerobic versus anaerobic environments, for example. Okay, next slide. Denitrification is very interesting. Essentially, with rare exceptions, the known marine denitrifiers are facultative anaerobes, meaning they prefer denitrifying environments, but they can switch to these aerobic respiration as needed. So the aerobes and the denitrifiers tend to strongly overlap. Denitrification itself is a modular process where some of the microbes contain all of the genetic abilities to go from nitrate all the way to dinitrating gas, but many do not, and many have just these pieces of that whole pathway and work in a way that's cooperating. There are a number of marker genes for each of these steps that are quite well known. Yeah, and I think that's all I'll say on that one. Okay, next slide. We're moving down the redox ladder and we're getting into lower energy environments, and to complement what David Burdige said, into a realm where there is much less known and understood, which means much more opportunity for fabulous discovery, but also more challenging in the interpretation. So manganese and iron reducers have the, they tend to be able to use a smaller carbon substrate spectrum because they're gaining less energy from these. They are performed by diverse microbes, but they have this additional challenge. Oxygen and nitrate is soluble. It can diffuse or be transported into cells. Iron manganese oxides are large particles that cannot be transferred intracellularly, at least when they're large. And so the markers that we tend to use for iron and manganese reducers is their ability to perform extracellular electron transport. That is the movement of electrons from inside their bodies to outside. And there's multiple pathways that have evolved for this, though there is some convergent, there appears to be some convergent evolution on this, where they tend to have these structural similarities, these beta barrels with porums and strings of cytochromes that enable electrons to move from inside to outside. There are at least 3 model organisms, the first 2 being Geobacter and Schwannella, which are unrelated, or distantly related, I should say, and have different extracellular electron pathways. Additionally, there's a gram-negative pathway for firmicutes that was more recently discovered. And this think here, instead of putting a fabulous picture of a microbe sending out extracellular electrons, this is from a Derek Lovely review showing the breadth of the extracellular electron transport abilities. Not all of this will be related to respiration with the environment, but it might be also electron sharing among distantly related microbes or unrelated microbes. Next slide. Okay, I don't think we can go any further without mentioning fermentation, because fermentation is the essential process that is providing the carbon substrates that are used by the sulfate reducers and by the methanogens. Fermentation, unlike respiration, is not the exchange of electrons to a terminal electron acceptor, but really it's the movement of electrons between organic carbon molecules. It generates less energy, much less energy than a respiration process. But it's used by many, many microbes. This very recent review found that fermentation was found in about a third of all prokaryotes for which they analyzed their metagenomes. They are far more distributed than I even realized, but the major groups where we knew there were a lot of fermenters still holds true. The firmicutes and the desulfobacterota, delta proteobacteria, have a lot of fermenters. Fermentation is a complex process. It can involve 300 different combinations of metabolites. In sediments, it generally requires that hydrogen be kept low. That is the product. And that's kept low by the interaction with the sulfate reducers and methanogens that are utilizing that. Okay, I think that's, so in sediments, additionally, atrobacteria tend to be an important group of fermenters that do increase in abundance through depth very typically. Okay, next slide. So sulfate reduction and the sulfate reducers, they are one of the key groups that frequently also have the ability for fermentation, presumably for dealing with fluctuating substrates in the environment. Sulfate reduction is one of those fascinating processes because, exactly as Talia was talking about, it's one of the processes that requires an input of energy in order to get out more energy. And that is specifically because of the pathway that's involved here. So I put up the genetic pathway. It's near the last step. Basically, sulfate is brought into a cell, and then it's an injection of ATP, which is energy, is converted to APS and then to sulfite and then to sulfide, which diffuses out of the cell, where the DSR-AB gene is used as a phylogenetic marker of the ability to perform sulfate reduction. A functional marker as well as a phylogenetic marker. There are at least 3 different pathways that have diverged in that form of the DSR-AB gene, one of archaeal origin, one of bacterial origin, and a different variation that runs in reverse such that the microbes are able to perform sulfide oxidation instead of sulfate reduction. In marine sediment, the DSR gene is actually vastly more found widespread in the tree of life, which I didn't fully appreciate. And most of them are not cultivated. But it's also true that in surface sediments, most of them are actually desulfobacterota, which are well cultivated in the labs. And there are other groups of secondary importance. And at deeper depths, other sulfate reducers can become important. The other point I wanted to make, though, is that even though these pathways are fairly conserved and that we have both, like the denitrification pathway, this one is also modular, and so some microorganisms may have part of the pathway to use some of those intermediate sulfur compounds. So if you were to use 5-sulfate or elemental sulfur for reduction, you wouldn't necessarily need that investment of ATP that starts this whole sulfate reduction process. To add minor complication, though, I will mention Campylobacteria in the next slide. They do perform sulfide oxidation, the reverse of this, by using the machinery for sulfate reduction, but in reverse. but they actually have a reductive, a forward operating sulfate reduce a DSRAB gene. So even these definitions aren't 100%, can't be translated 100% into our interpretation. Okay, next slide. So sulfide oxidation is performed by diverse microbes. A lot of the canonical sulfide oxidizers that we know from their sulfide oxidation genes, the SOX pathway. Many of them are in the gamma and alpha proteobacteria. There is also a well-known pathway, and then the light-dependent sulfur oxidizers have variations of this SOX pathway. There are also these microbes that are so-called gradient microbes or specialized sulfur oxidizers that are able to span the gap between the oxygen or nitrate zones and deeper sediment layers with sulfide. These include the very well-known, very visible gamma proteobacteria that include the giant sulfur bacteria, and now also the cable bacteria that I'm showing in that bottom picture there. You see threads of them connecting sulfur sediment grains. These microbes function very differently in terms of their biogeochemistry and in terms of how they gain energy and what they do. They also can strongly dominate in their environments. You can get major mats of Thioploca. You can also have very intense blooms of electrotherix, so cable bacteria, and those have strong influences on the whole biogeochemical cycling. For a nice paper on how this might affect, for example, pyrite burial, there's a nice paper by Hantsu et al. From 2023 that made some hypotheses on how their metabolism affects pyrite burial. Okay, next slide. I'm trying to turn the slide again. Methanogens, we're now at the bottom of that redox ladder. Methanogens are strictly among the archaea. They're strict anaerobes. And now they have a very narrow substrate spectrum. In labs, about 11 substrates have been found to be useful. There's generally 3 pathways for methanogenesis. The first two, the generation of the consumption of hydrogen acetate, the hydrogenotrophic pathway is slightly more energetically favorable than the acetoplastic, but both of those pathways compete with sulfate reducers. And are generally outcompeted by sulfate reducers that are able to drive down the hydrogen concentration in the environment. There's also a non-competitive pathway, for example, the cleaving off of methane from methanol. The distribution of the methanogens then is strongly influenced by the availability of hydrogen. I'm not going to elaborate on that. That's the tree of known methanogens in the environment. Okay, next slide. So methane oxidation, having this huge amount of chemical energy, then is able to be oxidized by everything else above it on the on that redox ladder. They are strongly able to be removed by methane oxidized by coupled with oxygen. These are all bacteria. They tend to be predominantly of the gamma proteobacteria, the alpha proteobacteria. They were discovered 100 years ago. Many of them were brought into culture in the 70s, and the organization of how we understand them, these type 1 and type 2, was derived in the 1970s and really is robust and holds up in our understanding of them today. They tend to have these membranes in them, and that couples the oxidation of methane with an electron transport chain. And there's a conserved gene involved in that, so we can identify these with gene markers. I mentioned Methylomirabilis. This is a different phylum completely. They are fascinating. What they do is they couple methane oxidation with nitrite, but they do so by generating oxygen from nitrite within their cells and then operating through the same pathways here. Okay, next slide. So methane oxidation by sulfide is probably very well known by everyone here. It's a reaction that requires a consortium of bacteria and archaea generally, and is the major pathway by which methane is removed from the sediments before it ever reaches the sediment surface at the sediment methane transition zone. Both members are able to complement each other in terms of driving this towards thermodynamic favorability. Okay, next slide. We're almost at the end. I've got 2 more here. But this is really key. The hydrogen concentration that is available strongly dictates whether these reactions are thermodynamically favorable in terms of whether they're driven towards methane oxidation or conversely methane production. I think there's an animate, or the next text box will come up if you click on it. So the reason this works is because the anaerobic oxidation, the archaea that are performing the anaerobic oxidation of methane are able to share with the sulfate reducers canonically hydrogen and acetate, though more recent understandings relate to elemental sulfur and direct electron exchange. But the reason I partly bring this up is to pose this additional question. In some eutrophic coastal zones, we do know that methane is able to break through from the sediments, that the sediment methane transition zone that is operating as a biofilter throughout most of the ocean does break down. And we don't 100% know why. And I do think this is an important question that is being addressed by different groups. Is it because the methane that's being produced is being produced by non-competitive pathways? I don't know. Or is it because there are very high hydrogen concentrations in sediment driven by high rates of fermentation? Okay, last slide. So it's my thought-provoking question. That's what that is. So big picture, and this is my summary. So the sediment microbiomes are highly structured and strongly shaped by the local conditions, especially with respect to organic carbon quantity and quality, and additionally by the electron acceptor availability, where electron acceptor availability makes it easier to interpret what we're looking at there. The nucleic acid archives may now supplement biogeochemical data to enable greater confidence. And certainly that's true of cryptic cycling. I would hope that that might also be true or becoming true of identifying forcing factors and giving us more confidence in our understanding of that. The interpretation of the sediment microbiome, though, is still is certainly steadily improving, but is still limited by our ability to interpret what is happening in the sediments. The more we look at it, the more exceptions we find, and the more possibilities for different pathways is occurring. Yeah, and that's all I have. Thank you very much.
Awesome, thank you so much for that. Okay, our last speaker of this session, we're talking about carbonate sediments, is Olivier Sulpis, and after this, we will have our coffee break, and then we'll go into our discussion.
Hello, everybody. Does somebody know where this is? It's not from an AI generator. From Marseille. So those are, what you see here is the national parks of the Calanque in Marseille, where I live. It's pretty slightly warmer than here. I feel pretty cold today. And those are carbonate sediments. So the nice thing with carbonate sediments is that first they're great places to hike in. They're full of climate archives, but also their dissolution is the ultimate sink for CO2. And so now in our current climate crisis, it's quite important to understand why they dissolve and how fast. So in this talk, I'll give you a very quick overview of the state of the art about calcium carbonate sediments, and then we'll share a few thoughts on the things that I believe are not fully understood yet. Okay, definitions. We have some formatting problems here. So just one slide of definitions. Just to make sure we're all on the same page here, PIC here means particulate inorganic carbon. So it includes a lot of things, including mostly actually calcium carbonate, which are defined by their formula. So it's everything that is solid and that contains calcium, C and 3 oxygens in it. And within those things, we have minerals. And minerals are actually defined in addition to what's in it, to the structure, to the crystal structure of them. And the main ones in the oceans are calcites and aragonites that you see small depictions of on the right. And that's it for definitions. Now, where do we find those calcium carbonate minerals? So here is a map that shows you their abundance. What you see here, if you look at the Atlantic, for instance, that they are mostly abundant in places that are quite shallow here on the mid-ocean ridge. On each side, because the pressure is really high, those carbonates tend to be dissolved. And it's mostly related to the fact that they are more soluble at high pressure. It takes less room to have those minerals dissolved than in a crystal state. And also we noticed something else, that when we get closer to the coast, the abundance decreases again. And that's mostly due to, well, a couple of things. The fact that they are diluted with other things, but also that in those coastal sediments, you have a lot of organic matter that is being respired, and that dissolves those minerals. So that's a traditional view of how calcium carbonates are distributed and why at the seafloor. So here you see a depth profile. Over 1/4 of the Earth today, the main component of the soil, of the seafloor in this case, is calcium carbonate. There are a lot of those minerals, and we find them everywhere up to the CCD, the calcite completion depth, which is the depth below which they dissolve faster than they accumulate. Then the sediments below that are mostly mud. And what's happening currently is that because we are acidifying the oceans, we're adding a lot of CO2 in it, while making them more acidic, and that will rise this CCD. And because of the way the oceans are made, there is a lot of seafloor area that is kind of around the CCD. So if it rises just by a little bit, you have a whole new area of seafloor that used to be places where calcite was accumulating, and that is now, there are now places where calcite is net dissolving. So it's really sensitive to small climate change. And what you see here is the overall chemical directions that describes their dissolution. Here CO2 is on the left. So when you add CO2 to water that contains calcium carbonates, you will drive them towards dissolution. And the fact that this dissolution happens neutralizes this CO2 in returns. So if we want to look at this in 2D, that's what we have. So the top map here shows you the dissolution rate. So if you multiply the units by 2, you have an alkalinity flux, the dissolution rate of calcium carbonates at the seafloor. And the map at the bottom shows you just the fraction of that dissolution that is due to our CO2, to anthropogenic CO2. So it's not a lot. It's mostly localized in very specific areas, here like the North Atlantic. But actually, if you divide this amount by the number of people on Earth, you have something like 70 grams of calcium carbonate. That's like a handful of white powder that we dissolve every day, every one of us, just by emitting CO2. So it's a lot, but it's nothing compared to the amount of CO2 we put in the first place, which is about 4 kilograms of CO2 per capita per day into the ocean. And we can look at this also in a 3D angle. So here you can tell that the seafloor topography has a high influence on how sensitive a given place is to a small CCD change. So what you see in blue are seafloors area that used to be below the CCD in the pre-industrial era or at the end of the pre-industrial era, and are still below the CCD today. What you see in pink are areas that used to be just above the CCD and are now below it. So you have a lot more pink area on the ridge here because it's not a very steep seafloor area. And here, if you check here on the trench, where the seafloor is much steeper, that CCD rise actually causes a much smaller increase in the area of seafloor that is now net dissolving. This has also biological or ecological implications. So this is from a study where the authors went to the Pacific and did ecological surveys on each side of the CCD. Here the CCD would be somewhere around there, plus or minus of uncertainty. And they realized that on each side you have two completely different ecosystems. So even organisms are kind of feel what the substrate is. And if suddenly we dissolve the carbonate sediments that they live in, they might not be happy anymore. And the conclusion of that study is that in addition to the chemical consequences, a rise in the CCD could have ecological effects and could trigger even extensions that would then be added on other perturbations that are also our faults, like deep sea mining. So in addition to perturbing the deep sea, humans are also affecting calcium carbonate in shallow environments. Here you see a typical warm water calcium carbonate sediment setting. We have different kinds of dissolution here. It's not all driven by the fact that seawater is more or less acidic. You have some part of this dissolution that's due to environmental parameters, so what's in your seawater around the coral reef. But you also have some dissolution due more to microenvironments that are just right next to the calcium carbonate minerals. And they would be more affected by things like respiration of organic matter. And you also have some other kind of processes that are able to erode or dissolve sediments. Here they group that into a term called into bioerosion. So this includes kind of physical breaking down of the coral reefs. And all of that together are what controls dissolution of carbonate reefs today. And the same group of authors did published in Science a few years ago a survey originating from many different field sites around the world, where they link the net dissolution of calcium carbonates in those sediments to the saturation state, omega, of the seawater around it. So omega, you can think of it as the potential for calcium carbonate minerals to dissolve. If it's higher than 1, it should dissolve. It doesn't mean that it will. If it's lower than 1, sorry, the other way around. If it's lower than 1, it should dissolve. If it's higher than 1, it should precipitate. And here, what you can see is that first, it's always higher than 1, but yet it dissolves quite a lot. And that's because this omega describes the chemistry of the water around it, or above the reef, but not necessarily of the water right next to the particles in those macroenvironments that could be much more acidic than the surrounding water because of respiration. So what they say is that in the future, as the omega of the overlying seawater will decrease because of ocean acidification, we will dissolve more and more sediments, coral reefs, and maybe at some point switch from net calcification to net dissolution of those sediments. Now about alkalinity. So one of the reasons why calcium carbonates seem interesting to a lot of people today is because they generate alkalinity. They can neutralize CO2, and in the context of carbon dioxide removal techniques, this gets a lot of attention. This is a study on blue carbon. So the fact that so the authors here used a sediment model to study what happens if you go from a bare sediment with nothing on it to the same sediment with a seagrass minnows or a healthy mangrove forest with and without calcium carbonate in the sediments. What they see is that in the case of the seagrass, I switched to others here. In the case of seagrass minnows, you have an alkalinity flux that appears even without calcium carbonate sediments present, and that's because of alkalinity generation mostly by anaerobic respiration. But when you add calcium carbonate sediments to on this place, this alkalinity flux increases a lot. And you have the same thing in the case of mangrove forests, and this is directly translated to a potential to remove calcium carbonate, to remove CO2 from the atmosphere. So this is some kind of a shortcut here. It's not because you have alkalinity added in seawater that it will remove CO2. You need to let it time to equilibrate with the air, but that's the idea. And on much longer timescales, we also see this relationship between calcium carbonate sediment dissolution and CO2 content. So here you can see how atmospheric PCO2 and the CCD are linked through time. So we are looking at very different timescales here. We're looking at the Cenozoic and then more recent times, the ice age successions here. You can see that whenever there is a strong PCO2 rise in the atmosphere, we have the CCD that is also rising, that is exposing a whole new area of seafloor to dissolution. that is generating alkalinity and neutralizing CO2. So that's a negative feedback loop. So we can see that in the sedimentary core. Here, looking at the core of the PETM. That's an event that occurred 55 billion years ago, in which about 1,000 petamoles of CO2 were emitted. And you can see that very well in the sedimentary core. You go from a sediment that is quite white, full in calcium carbonate, to something really dark. And today, how will the sediment core of the Anthropocene look like? That will depend on how fast can calcium carbonates react. Now, for more the perspectives, something I would like to focus on is the fact that we have different calcium carbonate minerals, and they are not only just living there side by side, they're actually interacting. So a way I like to think of dissolution or chemical reactions in general is something like this. Let's think of dissolution. Dissolution of calcite, for example, happens if you have a deficit in either calcium or carbonate ion in your seawater. That's the omega concept. If you don't have enough either carbonate or calcium, this will be compensated somehow by dissolution, for instance. And now, if you have too much of calcium or too much of carbonate for some reason, this will want to go away, and it will do so by precipitating some calcite. And the thing is that because we have different minerals, they have different solubilities because they have different crystal structures. They're more or less stable. They will have different balancing points on this. So if you have seawater that is at equilibrium with respect to calcite in a beaker, and you add, let's say, aragonite in the same beaker, it won't be at equilibrium with respect to aragonite because you won't have enough of either calcium or carbonate ion, and it will tend to dissolve. And that's the same thing for another group of solids here that I called magnesium calcites. They are like calcites, but as the name suggests, some of the calcium atoms in those minerals are replaced by smaller magnesium, which makes them unstable. That's what we know so far on the solubility of those different minerals. Here on the top, it would be highly soluble, here more stable. And here on the x-axis, you see the percentage of magnesium in the crystal structure. So magnesium doesn't go in aragonite, so it's a flat line. Solubility is here. It's more soluble than calcite, which is in blue here, pure calcite. And if we look at magnesium calcites, the one that can incorporate magnesium, roughly, apart from this little thing here, the more magnesium we have, the more soluble it is. But then we have three lines. So that's to tell you how little we know. We don't have the same solubility. It's from experiments. Whether the experiments were done on synthetic materials or biogenic samples that were either bleached or not. So there's a huge uncertainty here. But the idea is that for those percentages of magnesium, those should be the most soluble minerals. And who is producing those magnesium calcites? Mostly warm water organisms that I'm showing here. You have the temperature of the water they live in average, showing the color here. And so in a sedimentary system where you have all three, you would have magnesium calcite, aragonite, calcite. The one on the top would want to precipitate first and kind of protect the other minerals below. So that's also what we see in the watercolor. Here you can see the flux of calcium carbonates from the surface ocean to buried in the seafloor. It's more an open ocean vision where everything is vertical. And you can see what composes this flux. You have magnesium calcites in yellow that are quickly disappearing. They are mostly produced in the open ocean by fish in their guts. You then have a fair amount of aragonite that's mostly produced by theropods, the family of sea snails. They are everywhere at the surface ocean, but when you look at sediments, what's preserved, they are almost never there. So they have to disappear somewhere, and we think they mostly disappear here at the sediment interface. whereas calcites are much more abundant. They're mostly produced by copolypores, by foraminifera. They're easy to find with plankton nets at the surface, and are well preserved in sediments. So maybe what we should aim for when we try to describe sediments and the different calcium carbonate minerals that are in there is something a little bit like POC, like organic matter. You have not only one mineral at the top, but a lot of minerals over a very broad range of reactivity, some radioactive stuff, some much more refractory stuff. And in the end, the more you wait, the deeper you go, the more the reactive stuff goes away, and you're just left with the stable phases. So it's not really a view that's used yet. People like me spend a lot of time trying to find the right dissolution rate for a given mineral. We try to understand why, like the different mechanisms of this dissolution. But if we try to think like a modeler and say, okay, we want just a law that I can put in the model that works, maybe something like this would actually be quite convenient and quite accurate. Just food for thought. And the other big range of processes that I think deserve more attention is everything about the interactions between POC and PIC. So usually what you think of when you try to relate those two is that in a sediment or an environment where you have calcium carbonates, if you also have organic matter that's being inspired, it will generate some CO2 and dissolve the calcium carbonates. So you would think about this. When we have aerobic respiration, it acidifies the microenvironment, and it should lead to the dissolution of this. But it's a bit more complicated. If we stay on this side, the interactions from POC to PIC dissolution. If now your respiration is not in the presence of oxygen, but anaerobic, you will generate mostly alkalinity that could either lead to precipitation of PIC, or at least slow the dissolution down by some extent. And the other side of that diagram are things that I don't think are studied a lot, but the idea is that everything that affects PIC, the sedimentary environment, can somehow be reflected on the POC in the same environment. Think of things like this sand. It looks very permeable. If now everything dissolves, if you remove all the hard sand grains, and you just left with mud or softer organic substances, you would decrease the permeability. You could change the way water flows through it, and that could affect, in one way or another, I don't really know, the way POC gets degraded. You also have a lot of stuff that is absorbed on the surface of those minerals, and some of that stuff is called carbonate-associated organic matter, this DOC. And if the carbonate minerals are dissolved, then you release some of that food in the pore waters that could then be used to dissolve in more PIC, and you have a positive loop here. And you also have stuff related to what we saw earlier, the fact that PIC sediments are some kind of a substrate for some organisms. If the substrate goes away, the organisms would want to migrate or get extinct. And so, yeah, we have, I think, a lot of processes that goes really both ways. And final slide, why does that matter? As we saw first, there's a lot of PIC in the seafloor. If we just look at the top meters of the seafloor, there's almost as much in terms of molar quantity as anthropogenic CO2 that could be burned if we suddenly decide to burn all the coal and all the oil in the world. So it's an interesting reservoir of alkalinity, and it's subject to many anthropogenic influences, some that are going on right now, ocean acidification, but some that also could kick in if we pursue with all the CDR methods. The fact that it is quite sensible to ocean alkalinity changes and also to organic matter delivery. And we're talking either in coastal areas, blue carbon, but also offshore, things with afforestation that could represent a very large source of organic carbon and trigger for example, dissolution of remote pelagic sediments. And I believe that's it. Yeah, thank you.